Hello and welcome you all in the class of zoology. Today I will explain you about the electron microscopic structure of striated muscles. My dear student friends, as per your syllabus, there is one topic in the unit number 2, electron microscopic structure and chemical composition of striated muscles. So we are already known that muscles are the very important organs in our body which help us to give the mechanical strength to our body. So muscle cells are having the ability to undergo the excitable modes. Muscles having the ability to can transmit the action potential along their cell membrane. Then it's having the ability to convert the chemical energy into the mechanical responses. These are the very important properties of the muscles which is essential to the life of animals. Physiological properties of the muscles reveals us that they are having the ability to undergo the irritability or the excitability. Then they are having the ability to conduct the impulses. So they are having the ability to conduct the impulses from one place to another place. Contractibility is another most important properties of the muscles. As we are already known that the muscles they are undergo the regular contraction and relaxations. So as we are already known that structurally the muscles are having the multi-nucleated cells. Specifically the striated muscles or the skeletal muscles are nothing but that is multi-nucleated cells. As we are already known that there are the different kinds of the muscles which is present in the body of the organism. They are nothing but the striated muscles, cardiac muscles and the smooth muscles. Striated muscles are nothing but those muscles they are having the ability to attach with the tendons of the bones, and the cardiac muscles they are present in case of the heart of the animals. Then the smooth muscles they are present in the visceral organs of our body. Now there are three major types of muscles. They are nothing but the skeletal muscle, cardiac muscles and the smooth muscles. Now the striations which is observed here, in case of the muscles they are, that is in case of the skeletal muscles there is observing the prominent striations, whereas in case of the cardiac muscles there is observing the less striations, and in case of the smooth muscles there is not having the striations. Now the locations is the another important properties which is observed in case of the muscles. They are, in case of the striated muscles, begins and ends in the tendons, and in case of the cardiac muscles they are observed in the heart. Then in case of the smooth muscles they are observed in the hollow organs and the eyes. Shape of the muscles they are varied. Now in case of the skeletal muscles becomes the long cylindrical multinucleated. Cardiac muscles are the branch mono or the binucleated. Then in case of the smooth muscles, spindle shapes and the mononucleated. Comparatively, when we make the comparison between the three types of the muscles, at that time we will infer that the skeletal muscles are the long cylindrical multinucleated, cardiac muscles are the cylindrical branch and mono or the binucleated, and the smooth muscles are the spindle shapes and the mononucleated. Comparatively, anatomical functions in case of the, that is the three types of the muscle fibers, anatomical functions, connections. Now in this case, in case of the striated muscle, there is not having the any anatomical functional connections. In case of the cardiac muscle, there is having the anatomical functional connection in the form of the intercalated disc. Then in case of the smooth muscles, anatomical functional connections is observed or here in the unitary, and that is they are observed in case of the unitary and absent in the multi-units. Special features of these that is the three types of muscles. Striated muscles makes up the great mass of the somatic musculatures. Cardiac muscles having the pacemaker cells syncytial in functions, and in case of the smooth muscle, pacemaker cells syncytial functions unitary. Now inversions in case of the striated muscle is under the voluntary control. In case of the cardiac muscle under the involuntary controls, and in case of the smooth muscles under involuntary controls. Now here is the diagrammatic presentations of the three kinds of the muscle. Here this is the striated muscles, as here we already clearly understood that there is the alternate dark and light bands, so that in this case there is having the alterations of the actin and myosin filaments. Here is not having, in case of the cardiac muscle there is not having the such striations, and in case of the smooth muscles also there is a striation is also absent. Here in this diagrammatic presentation we clearly understood that the cardiac muscles, then there is a smooth muscles. Now here in case of the cardiac muscle there is a having the anatomical connection in the form of the intercalated disc. Okay, then here we clearly understood that there is an intercalated disc which is present in case of the cardiac muscles. As we know that in the muscles there is a having the muscle filaments, and the muscle filaments having the specialized type of the proteins. They are nothing but the actin and myosins. Actin and myosins are the two types of the protein which is present in case of the muscles. Here is the smooth muscles. Now these smooth muscles particularly observed here. Now the primary functions of the muscles are the having the ability to generate the force or the movements in the response to the physiological stimulus by the transducing chemical or electrical stimuli into a mechanical responses. Structure of skeletal muscles reveals us that structure is having the 40% of the body weights. They are having the ability to perform the locomotions in case of the animals. They are the striated or the voluntary under the controls. Then the muscle cells having the multinucleated cells. Skeletal muscles are nothing but the striated muscles. As we are already known that the striated muscles, they are attached to the tendons of the bones. Now there is having the muscle fibers, and the structural and functional units of the muscle is the myofibrils. Then the myofibrils having the alternated darker regions and the lighter regions. Okay, that is the alternate striations which is observed here in the form of the dark bands and the lighter bands. Now these dark and lighter bands having the actin and myosins. Actin and myosin is observed here. Now muscles are the epimysium, then fasciculus, perimysium, then it's having the muscle fibers, endomysium. These endomysiums are having the myofibrils, and the myofibrils having the myofilaments, and the myofilaments comprise the sarcomeres. Muscle fibers, as here in these ultra microscopic structures of the muscles, you will see that muscle fibers, they are covered by the sarcolemma, that is the outermost protective coverings. Now here there is having the terminal cisternae transverse tubules. Now it's form the reticular structure, they are known as the sarcoplasmic reticulums. Here in this case there is having the mitochondria. Now the myofibrils are present here. Now we are already known that the muscle fibers, striated muscles having the A bands and I bands, and in between these two A bands and the I bands there is having the lines, zigzag lines, they are known as the Z lines. Okay. Now sarcolemma are nothing but it consists of an inner plasma membrane and outer collagenous layers. In case of the muscles, they are having the invagination at numerous points to form the T tubules. It carries the action potential. In case of the cells, in case of the muscle cells, sarcoplasm consists of the myofibrils. Myofibrils are nothing but the numerous mitochondria lying parallel to the myofibrils. Sarcoplasmic reticulum are the important structure which is observed here in case of the muscles. Now it comprises the L tubules and it's a storage of the calcium ions. Now structure of the skeletal muscle fibers, as we are already known that the skeletal muscles having the ability to attach with the bones with the help of the tendons. Now it's having the epimysium. Then the epimysium comprises the perimysium. Perimysiums having the in between the numerous perimysium there is having the blood cells. Now when we observe the one, that is the perimysiums, there is having the muscle fibers, and the muscle fibers are having the, that is the myofibrils. Okay, so skeletal muscles having the fasciculus, muscle fibers, myofibrils and the myofilaments. These are the actual structural organization which is observed in case of the skeletal muscles. It comprises 10 to 18 mu in diameter each, composed of thousands of the myofibrils. Muscle fibers are 10 to 18 mu in diameter. Each muscle fiber composed of thousands of the myofibrils, and each myofibril is in turns made up of the myofilaments. Myofilaments comprises contractile myosin second and actin. Then it's also having the modulatory proteins in the form of the tropomyosin and troponins. Now sarcomeres is the units here. It comprises the fasciculus, and when we magnified the fasciculus, at that time we will infer that there is having the muscle fibers, and the muscle fibers are about 10 to 18 mu in diameter. Then each comprises the myofibrils, and the myofibrils comprises the myofilaments. Okay, myofibril comprises the myofilaments, and the myofilaments having alternate bands, that is the A band and I band, and in between the A band and I band there is having the zigzag line. These are nothing but the Z lines which is observed in case of the striated muscles. Now these myofibrils, that is the structural functional units of these myofibrils, are nothing but the sarcomeres. Now here there is having the clear cut diagrammatic presentation, that is the I band are nothing but the lighter bands, A bands are nothing but the darker bands. In the A band there is having the H zones. In the A band there is having the H zones, and in the I band there is having the Z lines. Okay, and the total unit is known as the sarcomeres. Total unit is known as the sarcomeres. Now muscle fibers comprises the myofibrils. Myofibrils comprises the myofilaments. Myofilaments comprises the alternate striations in the form of the I band and A bands. In the I band there is having the zigzag line, and in the A band there is having the specific regions. These are nothing but the H zones. Okay. Then when we magnified these myofibrils under the electron microscope, at that time we will clearly infer that there is a having the protein molecules in the form of the actin and myosins. Actin and myosins. Now here in the diagrammatic presentation we clearly understood that there is a darker regions and the lighter regions. Okay, I bands, then I bands again followed by the A bands, again A band get followed by the I bands. Now in the I band there is having the zigzag line, these are known as the Z lines, and in the A band there is having the, that is the specific zone, they are nothing but the H zones. Now here the myofilaments, myosins having the myosin filament are the special type of the proteins which is present in the myofibrils, and they, here there is a forming the special type of the bridge, they are known as the cross bridge, which is present specifically in the, that is the A bands. Then in the thin bands, as we are already known that the thin band and the thick band. Thick bands are nothing but the dark, and the lighter bands are nothing but the thin bands. Now in the thick band there is having the myosin, and in the thin bands there is having the actin filaments. So myosin and actins are the two important protein which is present in the myofibrils. Light microscopic structures of the muscles reveals us that due to the alternation of the dark and lighter bands, light bands, isotropic bands, lighter bands are known as the isotropic band or the I band. Here there is having the thin filaments. Dark bands are nothing but the anisotropic bands. Here these regions are known as the A band. They are undergo the bifurcations. Then H zones, they are present in the lighter, H zones are nothing but the lighter zone, and they are present in the A bands. Then the Z line in the center of the I bands is observed. Then in the A band there is also having the M line. Now the sarcomeres between the two Z lines, between the two successive Z lines, there is a basic units of the muscles are present. These basic units are known as the sarcomeres. Basic units are known as the sarcomeres, which is observed in the skeletal muscles. In this diagrammatic presentation, my dear student friends, we clearly understood how there is a differentiations of the I band and the A bands is observed in the striated muscles. Now I bands are nothing but the lighter bands, then A bands are nothing but the darker bands. In the I band there is having the zigzag lining. These zigzag lining are nothing but the Z lines. Then in the darker band there is having the lighter regions, and these lighter regions are nothing but the that is the H bands. Then here in this case there is having the straight line. These lines are nothing but the M lines. Okay. Now in between these two Z lines, now these are nothing but the structural unit of the striated muscles. They are nothing but the sarcomeres. Now myofibrils. Myofibrils is the structural units of the striated muscles. Now here we are already known that these myofibrils comprises alternate lighter band and the darker band. Okay, so they are also having the another names. They are nothing but the thin and thick bands. Thin and thick bands or the thin or thick filaments. Okay, they are associated with the protein molecules. Now the sarcomeres are the structural and functional unit of the striated muscles, as we clearly understood here. In this case, that is the actin and myosin filaments are present. We are already known here. Now actin and myosins are the two important protein molecules which is observed in the striated muscles, and they are associated with the another type of the proteins. They are nothing but the troponin and tropomyosins. Now the ultrastructures of the myofilaments reveals us that thick and the thin filaments. Myosins are 10 to 14 nanometer wide and 1.6 mm long, 1500 in each myofibrils, which is observed in the myofilaments. Myosin filaments made up of around 200 molecules of the myosins. Myosin molecules composed of 6 polypeptide chains, 2 heavy chains and the 4 light chains, a tail and arm and 2 globular heads. Arm or heads form the cross bridge. 2 hinge heads has sites for the ATP and actin binding sites. Now in this diagrammatic presentation we clearly understood that the myosin filaments having the head and the tail. Okay, so they are having the ability to form the cross bridge. Now here is the diagrammatic presentation of the myosin filament. As we are already known that there is having the heavy chains and the lighter chains. Now head regions, they are having the sites which is actually used for the ATP binding sites, actin binding sites, ATP binding site. Now here these regions, they are having the ability to form the cross bridge. Okay. A single thick filaments consist of many myosin molecules. They are present in the myofibrils. Shape of the single myosin filaments structurally, here there is having the head and here is the tail. Head and tail, these are the two regions of the myosin are observed in the striated muscles. Now head also known as the cross bridges. Okay, so here this part of the myosins having the ability to undergo the associations. Now part of the thin filaments, okay, as we are already know, thick and thin filaments are the two types of the filament which is present in the myofibrils. Okay, thick filaments are nothing but the myosin and thin filaments are nothing but the actins. Actin comprises the proteins. Actin comprises the proteins. They are nothing but the troponin and tropomyosins. Troponin and tropomyosins. Okay, thick filaments having the proteins in the form of the actin. Then these actins having the actin, troponin and tropomyosin. These are the three important protein which is observed here in case of the thin filaments. Actin filaments, they are about 7 nanometer wide and 1 mu long, extended on both sides of the Z line. F actin forms the double helix made up of the 300 G actin molecules. Molecular weight is about the 42,000 daltons. G actin has the active sites for the interaction with the myosin heads. Now the tropomyosins, troponin, tropomyosins, here there is having the 3 sites. Tropomyosin is about 40 nanometer length and the molecular weight of about 70,000 dalton, wrap around actin helix and cover active sites in the resting phase. Troponin made up of the 3 proteins TN1, TNC and TNT. These are the proteins, okay, which is having the ability to form the cross bridge. Actins, then troponin complex which is observed here, and tropomyosins are the filamentous structures which is present in the actins. Troposins blocking the myosin bridge sites with the help of the calcium ions. Myosin binding cell exposed by the calcium mediated tropomyosin movements. Other proteins are also observed here. Actin binds, that is the actinin binds actin to Z lines. Desmins are the another proteins, binds Z lines to the plasma membrane. Titin having the ability to connect the Z lines to the M lines and provide scaffolding for the sarcomeres, provide the elasticity. Dystrophin, dystroglycans and the sarcoglycans. Here is the diagrammatic presentation how there is a cross bridge is formed. Thick and thin filaments as we are already known here. Now here is these two Z lines. In between this there is having the lighter regions and the darker regions. Here there is having the titines, which is having the ability to connect the Z lines. Then here there is the thin filaments. Here is the thick filament. Thin filaments are nothing but the actin, and thick filaments are nothing but the myosin. Now alpha actin, which is having the cap Z proteins. Then troponins, which is having the ability to, there is having the multiple units here. Tropomycins, tropomodulins, which is also observed here in this diagrammatic presentation. Muscle fibers, as we already know, that the muscles, they are connected with the neurons, and the impulses which is received from the central nervous system, they are spread onto the muscle fibers, and these muscle fiber undergo the conducting the impulses, okay, and they are undergo the excitements and develop the action potentials, which is required for the working of the muscles. Sarcotubular system is another most important system which is present in the striated muscle. Tubules inward extension of sarcolemma opens to the exterior, contains a CEF, runs transfer to the myofibrils, transmit the action potential. L tubule sarcoplasmic reticulum run parallel to the myofibrils, terminate in the terminal cisternate, store calcium ions. Triads to terminal cisternate abutting the tubules. As we are already known that the action potentials are very important for the conducting the impulses from the central nervous system to the target regions. So action potential, which is observed in between the neuromuscular junctions, and here there is a muscles having the ability to conduct the impulses. In these diagrammatic presentations, we clearly understood that how the impulses they are conducted and how the muscle fiber undergo the excitements. Thank you so much for patiently listening these video lectures. Thank you one and all.